Good day and welcome to our topic 5 under our first module on antimicrobial agent. And we will proceed with our topic 5, that is about inhibitors of nucleic acid synthesis. So nucleic acid are molecules that store information for cellular growth and reproduction. So there are two types of nucleic acid. We have the RNA and the DNA. And these are polymers consisting of long chains of monomers called nucleotide. And the nucleotide, as we know it better now, consists of a nitrogenous base, a pentose sugar, and a phosphate group. So among the inhibitors of nucleic acid synthesis, we have antibacterial agents like inhibitors of RNA synthesis, and we also have inhibitors of DNA synthesis, and we also have antiviral agents like your reverse transcriptase inhibitors and integrase inhibitors. So antimicrobial drugs can target either RNA or DNA synthesis, as we spoke about. So the antimicrobial action of this agent are a result of differences in prokaryotic and eukaryotic enzymes involved in nucleic acid synthesis. And we know that prokaryotic transcription is the process in which the messenger RNA transcripts of genetic material are produced for a later translation into proteins. So we remember that we have that double-stranded DNA, then from there, there will be a formation of RNA, from the double-stranded DNA. So that's the transcription step. And the transcription process includes like your initiation, elongation, and termination phases. So antimicrobial drugs have been developed then to target, all these steps, whether they target the initiation phase or the elongation phase or the termination phase. So they act at different levels. Now let's look at the first group of these nucleic acid inhibitors, that is made of rifampicin, rifamycin, rifampin, or rifabutin. So these antimicrobials bind to the DNA-dependent RNA polymerase, and they then inhibit the initiation phase of the messenger RNA synthesis. So they act by inhibiting the initial phase of the messenger RNA synthesis. And they do have a broad spectrum of activity, but they are mostly used for the treatment of tuberculosis. So rifampicin is really reserved for the treatment of tuberculosis. Then we have the quinolones and the fluoroquinolones. We have nalidixic acid, we have ciprofloxacin, Oflox, Norflox, Levoflox, and so on. These antimicrobials bind to the A subunit of DNA gyrase, and prevent supercoiling of the DNA. Now, what we need to understand here is that the bacterial DNA is a very large molecule, is bigger than the size of a bacterial cell. Now, for that DNA to fit into a small bacterial cells, it needs to undergo a process called supercoiling of the DNA. okay, supercoiling that try to fit a very big, large molecule into a smaller size of a bacterial cell. And there are enzymes that played a role in that supercoiling, mainly the DNA gyrase. Okay, the DNA gyrase. So thereby inhibiting DNA synthesis. Okay, so quinolones have also a good spectrum of activities, and it's a drug that is mainly used to treat urinary tract infections. Then we also have, in addition to the antibacterial, we also have antiviral agents. Antiviral agent that inhibit nucleic acid synthesis. And here we have mainly inhibition of the reverse transcriptase, reverse transcriptase that play a role from RNA to DNA. Remember, we have DNA during transcription, we have DNA that is made to become a single-strand RNA. But then if you have an enzyme called the reverse transcriptase that turn our RNA back to a DNA. And those develop nucleotide or nucleoside analogs that look like the building blocks of RNA or DNA, but then deactivate the enzyme that synthesizes the RNA or DNA once the analog is incorporated. And some of those nucleotide or nucleoside analogs involve cyclovir. For example, it's a nucleoside analog and is effective against herpes simplex virus. We have zidovudine, that is AZT, is also a nucleoside analog used against HIV. We know that. And the lamivudine, that is also against HIV and approved also to be used against hepatitis B. So those are inhibitors of nucleic acid synthesis, and they play a role in the inhibition of reverse transcription. Okay, so let's try to look at our test one, just to recapitulate on things that we have studied. Which step of gene expression do rifamycin inhibit? Remember the group of rifampicin, rifamycin, we have spoke about. If you understood this lecture, which step of gene expression do rifamycin inhibit? Is it a DNA replication? Is it RNA replication? Is it transcription of DNA into RNA or transcription of RNA into DNA? So you can pause this video for 30 seconds and respond to the question. Okay, so if you remember very well, we say that rifamycin, they play a major role during the transcription phase, and it's mainly during the transcription of DNA into RNA. So the correct answer is C. The second test, which bacterial process or processes do quinolones and fluoroquinolones inhibit? Remember, we spoke about quinolones and fluoroquinolones. We spoke about supercoiling of the DNA, how to try to fit that big molecules into a small bacterial cell. And so which process do quinolones and fluoroquinolones inhibit? Do they inhibit DNA synthesis and RNA synthesis? RNA synthesis only? DNA synthesis only? Quinolones and fluoroquinolones do not affect nucleic acid. So pause your video for 30 seconds and respond to this question. Okay, so we can start by excluding some of the assertion here, like D. We say quinolones and fluoroquinolone do not affect nucleic synthesis. Nucleic acid synthesis, that is already wrong, so we can eliminate that. We know that we spoke about the supercoiling of the DNA, so we have nothing to do with RNA here, with quinolone, so we can also eliminate B. So by virtue of fact, we can also eliminate A because it contains DNA synthesis and RNA synthesis. So we remain with the correct answer, that is C. So it inhibits DNA synthesis, by interfering with the enzyme DNA gyrase. Okay. An extra test for you for this topic 5. Which of the following drugs is not a nucleoside or nucleotide analog? Which of these? We spoke about nucleoside and nucleotide analogs. Pause your video for 30 seconds and try to get the correct answer. Okay. AZT, didanosine, acyclovir, rifampin, decitabine. So already we can remove rifampicin because it's not in this group. We spoke about rifampicin, that is inhibiting transcription of DNA to RNA. So it's not a nucleotide or nucleoside analog. So which of the following is not? So it's D, rifampicin. All the rest are nucleoside or nucleotide analogs. Okay, so follow us as we move into our topic 6 under our module 1. We are going to discuss anti-metabolite. Thank you. Let's move to topic 6.